Hey guys, I'm here today to show you guys how to make a model that is going to be really helpful in getting a better understanding of mitosis and cell division. So instead of a model like you did for your biospheres, this is going to be a model that could be called an analogy. So you are going to be comparing the process of mitosis to something that you know better. And making that comparison is really helpful for clarifying concepts that might not be super easy to understand. So I'm going to go through an example analogy, and then after that you guys are going to each make your own analogy between the process of mitosis and something that you understand better. Okay? So to start out, I want to think about, what do I already know about mitosis? And so I might think, okay, I know that mitosis is the process of having one cell become two identical cells. Other stuff that I know for mitosis is that it involves DNA, and there are several steps which you learned about in previous lessons. If you remember, the word that was used as a reminder for the order of the steps, that was PMAT. And so these stand for prophase, metaphase, anaphase, and telophase. So this information is stuff that I think, oh, these seem like important parts when I'm talking about mitosis. So I want to include those things in my model. And then the next step is to think about, okay, what do each of these things do? So if I think about the DNA, I think, all right, what's the role of DNA? And I know it's to kind of give instructions to the cell. So because of that, this starts giving me an idea of where I can go with my analogy. So I know DNA gives instructions, so what other things do I know of that give instructions? Okay, and so the first thing that comes to my head is directions for like building furniture. So building like an Ikea table, let's say. When you get the box from Ikea, inside that box, there's this piece of paper with a bunch of directions that tells you, like, okay, how am I going to put this together? So we can think about the DNA being kind of like those directions. And then if the DNA is like those directions, the thing that the DNA is making, or what the instructions are telling you how to make, would be the final table. So you can think of the table being like the proteins that get made based on the directions of the DNA. Oh, that's spelled wrong. Oh well. And you can think about the actual instructions as being the DNA. So now I want to start thinking, all right, I'm going to erase this. I have a couple of these pieces, but I want to get into it with more detail. So I'm going to erase everything, and I'm going to start with the DNA. So when IKEA first designs a table, and they want to tell us how to make a table, I'm sure they have to write up the directions, and they have those on some kind of computer, like at IKEA headquarters in Sweden, or wherever. So the directions would be on the computer in Sweden. Okay? I know this is a really good drawing. So I would say this is equivalent to my DNA. And now this is how we're going to make one table. But now I think, okay, I want to make a bunch of tables, or I want to have everyone be able to make a table, and I want it to be exactly identical to the table that we made based on our original instructions. So that means we're going to need to make a copy of the DNA. Okay? You can think of that as being like the printed out directions to make the table. And then after we copy the DNA, then we're going to keep on going and we're going to think, all right, what's the next step? So now I'm trying to think of something. The DNA has been copied, and now we're getting into the process of mitosis. And if we think about the first step of mitosis, we remember that's prophase. So in prophase, we might say, oh, okay, well, that's when the DNA starts getting like packaged in a way that makes it easier to make a copy. So maybe we're thinking prophase is when you get your Ikea box and you get it home and you open it up. Okay? So prophase is opening it up and taking the directions out of the box. Okay? Then the next step after prophase, remember PMAT, is metaphase. So if we think, all right, what happens in metaphase? We remember that the DNA starts lining up in the middle. So this is kind of like organizing the parts for the directions. So maybe we think, all right, we have our directions out, and now we're going to start taking the parts of the table out of the box and laying them out in a way that we can use all of them. Okay, and there are some like screws in here or nails. I don't know. I'm not sure what goes into an Ikea box. Okay, so now we've organized stuff like we do in metaphase. And then we start pulling the stuff apart during anaphase. So now we're gonna like organize it a little bit better. So maybe we start to put a leg onto the base or the tabletop. So the table's on the ground, we have it upside down, and we start putting those legs on. Then telophase is when you put all the directions back where they came from. You're starting to get ready to have two brand new different cells. So telophase, maybe we're going to take our table, and it's all built now, and we put it upright. Okay, and here we have an analogy for the process of mitosis. Now, the important thing to keep in mind is that this is in no way a perfect representation of mitosis. There are lots of issues with it, like, well, we're talking about organizing the parts here, but really we're actually organizing the DNA, and we said the instructions were DNA, so like that's very confusing if we think about it in more detail. But as a base level comparison, this is a pretty accurate comparison between the process of mitosis and how to build an IKEA table. So it's fine that it isn't perfect, and I don't expect your model to be perfect either. But this is just kind of the general idea to really start getting you thinking about those steps and what goes into each step in this process. Okay? So next up, you're going to make your model, and I look forward to seeing what everyone comes up with.